So I'm here to present the SciLifeLab Pandemic Response and Preparedness, sort of all the efforts that SciLife has all the efforts that SciLifeLab has done during the COVID-19 pandemic, both sort of during the response time, but also for future preparedness. And this is also where Jens and co-workers fit in really well, because they have been part of all these efforts. As you may know, the pandemic can be divided into different phases. And important phases during the pandemic is during the alert phase and pandemic phase, where basically infection rates, morbidity or mortality rises dramatically. During this phase, it's very important with response. So this is kind of the response phase of the pandemic, where you need to take care of the acute measures of the pandemic. After that, you enter something called the transition phase before you get back to the interpandemic phase. SciLifeLab has been involved during all these parts of the COVID-19 pandemics, because in the beginning, there was a lot of focus on response. This is often called also wartime. This part of the pandemic where action needs to happen immediately. After that, SciLifeLab has also been very involved in the peacetime, so this transition phase, and is now planning more for the interpandemic phase, where it's more towards preparedness for the next pandemic. If we travel back in time to just after WHO declared COVID-19 a pandemic, SciLifeLab launched 4 main initiatives, and this was already in March 2020. The first of them was a Wallenberg Foundation-funded program for SARS-CoV-2 testing. And this was, of course, very important to get the testing going so that we could get a good idea about what was happening during the pandemic. The Wallenberg Foundation also initiated, together with SciLifeLab, a national COVID-19 research program. And you'll hear more about that in a little while. The Swedish Research Council also commissioned SciLifeLab to coordinate something called the National COVID-19 Data Portal, which was important for open data sharing during the pandemic, so that research data, etc., could be shared rapidly and swiftly. And SciLifeLab also decided that infrastructure should be prioritized for COVID-19 projects. So the SciLifeLab and Wallenberg National COVID-19 Research Program was launched then in March 2020. This was a broad launch, and basically first 285 proposals came in. There were 24 different topics, and in the end, 67 projects were funded. And these were in 9 research areas, as you can see here over to the right. And drug discovery and repurposing of drugs was a pretty big part of this, so 15%. But of course, also other parts of research were funded as well. This continued with a second phase. In the second phase, an additional 34 projects got funded. And again, drug discovery and repurposing of drugs, which is relevant for today, got a big chunk of the money. But as you see here over to the right and to the left, that quite a broad spectrum of different pandemic response research was funded. So already then, quite early on, SciLifeLab had then started to get sort of quite a broad network, because all of these, the ones that were funded, there was highly, that collaboration and team-based science was a key aspect of all of this. And many of the funded projects actually had collaborations going on in between. So then when we reached 2021, the Swedish government decided to improve sort of the pandemic preparedness of Sweden. And again, SciLifeLab was contacted for this. So already now, there was already a link between the national infrastructure of SciLifeLab to healthcare, national research programs, and there was already a data portal that had been created. So this was perfect for SciLifeLab to build upon, to basically to make a new capability called the Pandemic Laboratory Preparedness capability. Here is basically what the research proposition that the government sent out looked like, where they basically said that the government supports SciLifeLab with extra funding to build better capabilities to handle future pandemics. So now is when you move from more response to more pandemic preparedness. Importantly, this was supposed to be done, or had to be done, in consultation with Folkhälsomyndigheten. And also that this was sort of supposed to lead to research being performed, technology developed, and education to happen within the SciLifeLab umbrella. And that would complement and support governmental agencies and local authorities. So quite a slim organization was decided for this pandemic laboratory preparedness program, where Staffan Svärd is the scientific co-director and Alice Solazzo is the coordinator. These two were in the organization board already from scratch. I was a late addition later on, Ulf Rybacke as a co-director. The vision of PLP was to always have the research community as the core. The research community should basically be the core of all actions done within this program. And this core of the research community was supposed to, or is supposed to be sort of supported and surrounded by the SciLifeLab infrastructure. And what was really important here as well was to integrate basically this research community into the infrastructure of SciLifeLab to really increase the sustainability of this program. As the next layer of this vision was the data portal, which would basically then enable open and rapid sharing of data and findings. And then, of course, this was done in consultation with healthcare and governmental agencies, and then also outreach to industry and the public sector. So pandemic laboratory preparedness is just one of the three capabilities within SciLifeLab. Here you also see precision medicine and planetary biology. And these basically act like thematic cores that are joining sort of the national research community with the national infrastructure of SciLifeLab, together with the data-driven life sciences, or DDLS. So these three capabilities serves as nodes, basically thematic nodes for research. So when the PLP program was to be built, it was decided that the absolute bulk of the money should go straight to the research community. And this was done through competitive grant applications. And there have been several calls during the years. In the beginning, there were 21 units, eventually it turned into 22 units that got funded. And these have gotten the chance to get renewed grants over time. And just recently, we closed the last call, which was called PLP Test, where 10 of the already funded programs are suggested to get funded again. So these 22 PLP units are spread out on a national basis, pretty much everywhere, but you can see that there's a bit of a focus, or there are foci around where we have our SciLifeLab sites. As you see here to the right, that the SciLifeLab capabilities that were sort of built up here, that was a big focus on sequencing efforts, both in terms of diagnostics, but also for identification of novel variants of viruses, etc. A BSL-3 network was being created because that was very apparently that it was needed in Sweden. There was prior to this, was very little communication between different BSL-3 facilities in Sweden. There was also a lot of immunomonitoring, surveillance, etc., environmental sampling and processing that got sort of involved in this. Also key to this program was to involve the clinical microbiology labs, spread all over Sweden, from Norrland up in the north to Lund down in Skåne. And this was all the time done together with the governmental agencies, Folkhälsomyndigheten, FOI, and SVA. And more recently, we've also started to collaborate more with international organizations such as ECDC, Covarnet, and IVI. So this is the current status with 22 units that are funded by the program. And again, this is a what was highly appreciated was if these units would collaborate in between. And that has really turned out well. So they collaborate all over the borders in relationship to the projects that they have. And partly due to the SciLifeLab efforts together with Wallenberg Foundation, etc., has led then to quite an impressive number of publications related to COVID-19 from Sweden. As you can see here, in May 2023, there were over 3,000 COVID-19 publications that had been sort of generated. And a big bulk of these actually had funding from the PLP program. So one can easily say that the PLP program is integrated into the SciLifeLab infrastructure. And over here to the left, this is quite a busy slide. So if you only focus on the left side, you'll see these little stars or snowflakes or viruses, depending on how you look at it. You can see how different projects are sort of integrated into the SciLifeLab infrastructures. And if you focus down here, you can see that here you find drug discovery and development, the platform that is basically hosting today's seminars. And that is also where Jens Carlsson and co-workers have been sort of integrated during their work to try to design and develop new drugs against COVID-19. And with that, I pass on the word to Jens.
And before that, there is some room for questions. If you would like to raise that, please raise your hand or write in the chat forum. If I don't see anything, we can take questions later on as well. So 15 seconds during the break here. You must have had a very clear presentation, Ulf. Yes.
Or they're saving the questions for Jens.
Yes.
The hard ones.
Okay. Take it on then. Yeah.
I will share my screen.
Start your video.
Start my video. Hello. Okay, now I will share my screen. Okay, thank you for the opportunity to present the journey that we've made in our project to discover main protease inhibitors. When the pandemic started, or before the pandemic started, my group has been interested in family of receptors, and we had no interest in viruses. We're just trying to understand how proteins work and how molecules interact with the receptors in the body to understand how they function. Doing that, we develop computational methods to predict how molecules interact with receptors at the molecular level. And we've always had a very tight interaction with experimentalists wanting to get into the test tube to test our computational predictions in the lab. And when the pandemic started, I was at first not interested. I thought this would be over quite soon and we didn't have to worry about it. But I realized that as many labs closed down, we should also focus on understanding the virus, try to find inhibitors using the methods that we had developed. So we started writing on the grant application, and here you see two of those that were submitted to the SciLifeLab call. One was from my group and Helena Danielson's group, trying to use a method called fragment-based drug discovery. And at the same time, Christian was trying to put together a larger initiative that could support several research groups interested in developing inhibitors. I realized, going back to that presentation, that maybe we were overly ambitious. I said that we can do this in 30 days. And I still think that is not an unrealistic goal. In the future, we will be able to do this. But for now, I have to apologize to the Wallenberg Foundation and say that it was more like 3 years. So we should remember that if we already had an antiviral drug in 2020, a lot of lives would have been saved. You know, someone's grandmother, people's family members. So it would have paid off to work on pandemic preparedness and develop antiviral drugs. And still now, with the vaccines being available, we know that we will need antivirals both to treat patients that cannot be treated with the traditional vaccines, and we will also need it for the future. And as you can see, you know, 6 million people actually died from this disease. In the beginning of the pandemic, there was many researchers and pharmaceutical companies that focused on something we call repurposing, that you can take an already existing drug to treat patients. It's a brilliant idea. It's the first thing we should do. But we know now that these efforts were actually not very successful. So here you see a president that you might recognize. He suggested to inject disinfectant into people in order to cure the virus, which does not work. He was also treated with remdesivir, showing an antiviral drug, but not developed for COVID, showing that, you know, people were actually using everything they could to try to cure the disease. But now we know afterwards that many of these drugs actually didn't work. When we see the evaluations of mortality or hospitalization, most of the repurposed drugs actually did not have any effect. I should say remdesivir, it goes a little bit back and forth, but so it seems to have some effect, but many of those suggested do not work. And we know now, I've realized now that 30 days plan is not going to work. So we need to start working well in advance before the pandemic shows up to develop the antiviral drugs. Now, after all the TV news, we know that there were SARS-CoV-1 in 2002, MERS-CoV, which was a very deadly virus, 2012, and SARS-CoV in 2020. So if you can do the math, you add 8 or 10 years, and we will be back fighting these viruses. I was also surprised to learn during the pandemic that a lot of viruses do not have vaccines or antiviral drugs, actually a surprisingly small number to me. So we need more research in this area. Going back to SARS-CoV-2, the whole world started focusing their research on this protein, on this virus, trying to characterize the different proteins and components of this invisible enemy. We got particularly interested in the main protease, which is crucial for viral replications. It cleaves proteins that are produced by the host cells. The strategy has been successful before, that you can target enzymes such as proteases, for example, against HIV, for which now patients can be saved. And today, I think you don't die from HIV. You're treated with a combination of drugs very successfully. It's also strongly conserved, so we can think that if it works for SARS-CoV-2, it could also work for other past viruses and those that will visit us in the future. At this time, there was no, okay, there's a little bit of problem. Let's see. Okay, I have a little problem on the top of the slide, but it's not very important. What I want to say is that there actually is a drug now targeting this enzyme. It's called Paxlovid, and the molecule that targets the enzyme is called nimetravir. And that's great. It came out in 2021, but we know that this drug has some potential drawbacks. It, for example, is metabolized quickly, need to be given together with another drug, which complicates treatment of some patients. And we also, of course, worry about resistance mutations, which are very common in treatments of viruses. So when we were starting the project, none of these compounds existed. But even if this new drug exists now, we believe we need additional ones. So we were very happy to see some of these grant applications that we sent in to be accepted by the KV SciLifeLab research program. And then Christian started to set up how this infrastructure would work, because we had several different groups interested. We wanted to use several different types of technologies. One was DNA-encoded library, a very new and exciting strategy to identify inhibitors. my group had been working on virtual screening and took the lead there. Helena Danielson had long time experience from working on fragment-based drug discovery and worked in this area, whereas Lindon and Anja had worked on a group of inhibitors called peptidomimetics and worked on that. We also got libraries from pharma companies that we could use. And all these projects were running in parallel. Some were sharing data between them, others were not. But SciLifeLab enabled us to run all these projects at the same time without sort of interfering with each other if we didn't want to. We also got access to a lot of assays. So working in academia, I know how difficult it is to recruit collaborators and carry out many different types of experiments. It's like juggling too many balls at the same time. Now we could put this all in the same place and interact with SciLifeLab to test the compounds. We were also able to interact with virologists, such as Lundqvist and Lennerstrand, to test in infection models. And during the project, which was the most exciting thing, we were able to start collaborations with lots of other national and international groups to set up a possibility to test extensively at viruses and with MAX4 to solve molecular structures of inhibitors together with the enzyme. Over time, we were closer and closer with a core team consisting of our group, Helena's, Lindon, and Anja. And I will present some of the research that were carried out in the virtual screening and fragment-based lead discovery projects. So our groups work on virtual screening, how you can use computers to try to design small molecule drugs. And the idea is that maybe we can do it faster and cheaper, but also that we can cover a larger library than is possible with traditional screening methods. For example, at SciLifeLab, we have about 40,000 molecules that you can screen experimentally, whereas in the computer, we can screen millions to probably even billions of molecules. So we just developed several types of strategies where you use a very simple technique where you take each molecule and computationally try to fit it under the surface of the protein. This is called a structure-based drug design, and we use that together with simulations and artificial intelligence to try to design molecules using libraries that reach the billion scale level. This research was funded by the ERC, but we could take this technology and then apply it to the main protease. Finding a drug is like, looking for a needle in a haystack. It's been estimated that the number of possible drugs is 10 to the power of 60. And that's more than the number of atoms in our solar system. So you understand this is a very difficult problem. And the number of molecules we have access to is even smaller. There are a few thousand approved drugs. There's about 10 to 20 million molecules that exist on our planet today that you can order. But what has happened in the latest couple of years is that we've gotten access to what they call make-on-demand libraries. Molecules that companies think they don't have them, but they can make them. And this is a great opportunity for computational chemistry and overall medicinal chemistry to faster move into testing molecules for activity. So here you just see an example of how the number of commercial molecules has grown over the years, starting in 2005 when I started my research career in this area. And in around 2020, when we started, there were about 300 to 400 million molecules available. Today, it has reached about 3 billion. And Enamine, a company from Ukraine, can make about 30 billion compounds in their lab, send it to you in 4 to 5 weeks at a very low cost and high success rate. So this is, I think, a great opportunity for projects where you need to progress fast. So back to the target, the main protease. Researchers quickly solved crystal structures, share that very rapidly with everyone around the world, so everyone could work on inhibitor discovery. The compound you see bound here is an inhibitor, but it's very weak with a Kd of only around 2 micromolar, and it's a weak compound in cells. It can't be used to treat patients, but it's a good starting point for us to develop compounds. When we carry out projects, we like to have of course, the idea to develop an inhibitor, but we also want to develop strategies or compare strategies to develop molecules. So in the first screen we did, we screened 200 million just random compounds from the large chemical libraries. So this enabled us to screen a lot of different molecules in a very rapid way. But another approach would be to start from a small molecule that you know binds to one part of the binding site and expand that into a different pocket. This we refer to as a fragment-based approach. And there you can need to screen fewer molecules, but it should be a good idea because you know that this gray molecule here binds to part of the binding site. Maybe you can just quickly optimize it. Here you see an example of the ultra-large virtual screen that we performed. About 235 million molecules were screened, and we scored 233 trillion complexes on the national supercomputer clusters. This is not possible to carry out at home on your iPad or your home computer, but access to the Swedish supercomputers allowed us to do it. And they also gave us extra time on the centers in order to perform the screen. And this was carried out by my PhD student, Andreas Luttens. So here you see the binding site with the compound that we had access to via crystal structure. Then we remove that and add other molecules that we try to fit into the binding site of the enzyme. And here you see some of those that were highly ranked by the scoring function that we use. Among those, we selected 100 molecules and ordered those in June 2020, about 1 to 2 months after the project had started. Testing those, we performed SPR, a binding assay, to see if they would bind to the protein, and an enzyme inhibition assay to see if they could inhibit the protein. And all of these assays were performed by the SciLifeLab Drug Discovery and Development platform. Here you see 3 of the inhibitors that we discovered. Those had activity in both the binding assay and the inhibition assay, so we think these were solid hits. But you see that they are very weak. At 50 micromolar, they only inhibit about 50% of the activity, which means that these are very far from being anywhere near a drug. But they're a starting point that we can use to develop better compounds. Here you see some experimental assays from biosensor to determine the binding affinity, as well as a crystal structure that Vladimir solved for us, working late nights, max 4. Here you see the model that we had in green, overlaid with the experimental data obtained after. And you can see that our computational model was agreed very well with what we had predicted. And this was great for us because working with the receptors, you almost never get structures of your design molecules. So I got very excited by the fact that we so quickly could get access to experimental data. In the second screen that we did, we performed fragment-based screen. And this was based on the work carried out in the UK at the Diamond Light Source. When the pandemic started, already in February 2020, they performed a fragment screen trying to find very small molecules that bind to the surface of the protein. And all of this data was quickly shared with the community so we could take on this challenge. The fragment that we decided to optimize bound very weakly. It would not even be detectable in an enzyme inhibition assay, and maybe actually we could only detect very weak binding. But using that template, we could define a general structure or an idea of how a molecule could look like that bind to the target, search in very large libraries of billions of molecules, and identify the 2 million that we thought could bind to the enzyme. Here you see our screen that we performed. And again, you see the original inhibitor that we had access to. And I will now show a few of the fragments that were discovered at the Diamond Light Source. The fragments often bind to small sub-pockets of the binding site, and you need to optimize them to grow them into other pockets in order to get a potent and selected inhibitor. Here you see one of the fragments that we found particularly appealing, and another one just dipping down in another site. And what I will show now is that we take this fragment, then we search for molecules in the large libraries that we can get access to and screen those. So you see these molecules all have something in common. They all have the core to the left the same, but have other groups growing into the other pockets. And here we again selected around 100 compounds and ordered those in 2020. From the virtual fragment screening, we also identified several hits. Here you see again SPR and enzyme inhibition assays, and 5 molecules bound to the target and also inhibited again very weakly. You can see that they all have something in common that I've circled there in red, but the other groups interacted in different ways with the enzyme active site. Here you see the experimental data as I showed in the other case. And again, we were surprised, happily surprised to see that our design agreed very well with the experiments carried out at MAX4. So if we compare the two strategies, which was our research question, we could see that the two approaches got the same hit rate. We tested about 100. We found about 1 to 2 new scaffolds, and they have similar potencies. This surprised me, actually disappointed me. I thought the fragment-based virtual screen would work much better and gives us ideas or directions on where we need to improve our research. But we are overly very happy that our predictions agreed with the crystallographic structures, which enabled us to continue to optimize the compounds. Here you see one example of how we work when we optimize molecules. And then we take 2 fragments, 2 small molecules that we have identified, and try to merge them, identify features of both molecules that could lead to an even better inhibitor. And here you see we can optimize a molecule for around 20 micromolar, very weak, into 2 micromolar, where you could possibly start to see inhibition on the cell level. Again, we use the commercial libraries. We order hundreds of molecules at the time, enabled by the generous funding by the Wallenberg Foundation, and allowed us to move on very quickly. We've now enabled optimization of these to around 200-300 nanomolar. Here you see another case of optimization where we take a molecule and we modify it to optimize interactions with the enzyme. And here we found something around 600 nanomolar where we could show that we have an antiviral effect in cells. And again, the crystal structure shows that the molecule interacts with the target. In the hit optimization phase, we explored the commercial libraries together with synthesizing molecules by ourselves. This is the series that is now, in a sense, exiting from the platform where we reached the furthest. So we started modifying the left part, the blue part of the molecule, systematically changing the groups to optimize potency. Then we took the other side, keeping the left side constant, we optimized the right, red side, and again achieved about micromolar potency. The problem here that we find working in these large commercially available chemical libraries is that we get stuck. So we were needed to engage chemists. So Dewey in my group synthesized the molecule that combined the best parts of each optimization path. And we were the first time able to reach sub-micromolar potency, which enabled us to do a lot more experiments on cells. Then we actually submitted our manuscript, and we got a lot of some criticism saying that, you know, your molecule is not very potent. 300 nanomolar is not very good. But at the same time, we were synthesizing molecules and trying to use the structure that you see here, solved at MAX4, and identify the pockets which we need to optimize in order to achieve better potency. And we managed during this time to optimize by adding one single atom at the right place into a 77 nanomolar compound. It was high affinity, it was selective, it didn't bind to other human proteases in the body, which is very important, of course, to not get side effects. And in particular, it had a low molecular weight. I think it's only like half the size of the compound that later got approved from Pfizer, which is a good starting point for further optimization. Here you see experiments performed by Åke Lundqvist and Johan Lennestrand's labs, where we looked at effect on cells. When you develop the molecule, you're afraid what's going to happen on cells. You only test them on the protein. If the molecule doesn't cross the cell membrane, nothing will happen. But we found that our molecule, this small molecule, will easily access the inside of the cell and inhibit it very potently, which you can see here on photos of these experiments. We could also compare to experiments performed together with, or in parallel, we performed experiments together with the Pfizer drug, and we could see that our compound has actually had comparable activity to the one that had been developed and gone into clinical trials. Another important question is, what about the other SARS viruses? If we can inhibit those, maybe also we can inhibit other viruses that will come in the future. And if there are outbreaks of the old viruses, we could also treat those. And here you can see that the molecule, our compound 19, that was very potent, also inhibited the 3 other viruses, which we did in collaboration with researchers at KU Leuven and Leiden University. So again, this showed the fantastic collaborative efforts that we could interact not only within Sweden with SciLifeLab and other research groups, but lots of other groups were willing to contribute to develop new compounds. So the support from DDD and KV really enabled us to progress faster than we ever done before. We were buying sets of molecules of 10 and 20 of our designs. Evaluating the compounds would take 2 to 3 months. Here we worked with iterations of weeks instead of months, which was very exciting. So overall tested about 400 compounds, determined several crystal structures with intercellular models in a way we've never done before. The paper was published. It has received great attention. It was one of the most viewed papers on JACS for about a year. But at the same time, we started thinking we should continue this project. And we filed a patent application and submitted new applications to the SciLifeLab COVID program. Here, Helena Danielson set up a team together with me, with Anja and Lindon, who are medicinal chemists, in order to further progress. And we created something that we called cold therapeutics, which was an idea of how we could potentially commercialize this invention. At the same time, we also got money, or actually Christian got money to set up more development of COVID or antiviral molecules, which enabled us to get into the SciLifeLab drug discovery program as an in-house project. So this, we were funded, and the SciLifeLab took us in, and we started what we call the COLD team, or coronavirus lead discovery, which is Anja and Helena had a lot of experience from antiviral drug discovery. Also, Helena had experience from the business development part side of things, which is also super important. Andreas and I were scratching our heads in front of the computer, and we had a team of 3 medicinal chemists, Dewey, Mohammed, and Tanusha, who made molecules, supervised by Anja and Linda. At the same time, we were meeting every week with the Fredrik, sometimes Christian, in order to coordinate all the assays that SciLifeLab was running. Only the management of the compounds, around 1,200 compounds. If you store that in your freezer in the back of the lab, it will never work. So just having the infrastructure to send compounds to different places, to the right place at the right time, enabled us to continue to develop the molecules in a really nice way. So in this project, which has been as long as the first part of the project that I presented up to the publication, we worked on optimizing different parts of the molecule. So we looked at the S1 pocket, the S2 pocket, and here it's not only important to optimize for affinity, which has been the only thing I've done before when I didn't think about the big picture, how to get to the drug. Here we need to also worry about the ADME properties. Is it soluble? Does it permeate the cell? Is it metabolically stable so we can test it in mice and rat and dog and eventually in humans? And here, SciLifeLab, in particular, and the ADME part supported us so we could progress it toward something that could possibly become a drug. We worked with the DMDI to do the antiviral assays, and Max4 continued to support us by determining crystal structures. So here we have this design, make, test, analyze cycle going on month after month, week after week, continuously optimizing the molecules that we had discovered. In total, we made about 450 compounds belonging to the series that this hydantoin represents. We tested those 250 in enzymatic assays. The best one also went on to biosensor assays, where we could really see how tight they were binding to the protein. We tested them in antiviral assays, and as well as continuously performing the ADME assays to see, are we modifying the properties? So we're going in the right direction, because these molecules were initially quite hydrophobic, not very soluble. They will not make good drugs. So we needed to optimize them. And as part of this, we've also submitted 3 patent applications. That's actually the end of the presentation. And as you can see here, there's a really long list of people that contributed to this project. On the academic side, we started off working kind of independently, became a strong team with me, Helena, Anja, and Lindon. We were always supported by this great middle team here, led by Christian and coordinating. There's just too many names to name all of them. We're just super grateful for everything you've done. We work with the Chemical Biology Consortium. They receive the compounds, package them, go to different places. Also fantastic. And we had many collaborators from the MAX4 in particular, but also a lot of other facilities, such as the Protein Science Facility, contributed to our project. And when you're doing all these experiments, you also have to keep an eye on what's happening in the world, what are other people developing, how can we manage to patent our compound well in time, how do we find a partner? And the business development, first via UU Innovation, then together with UU Invest, and UU Innovation hooked us up with the right people to work with as external experts, how we should navigate. We finally managed to do something that was impossible, to partner out this project. And that's why we're here today. So thank you to everybody. If I have forgotten everyone on this list, you should come to me and say that, and I will buy you a beer, which you can drink together with me. And we can talk about how great this has been. So thanks a lot.
Okay, thank you very much, Jens. I have right now difficult to see raised hands. Do you see any? So are there any questions to Jens or Ulf that you would like to ask? I'm looking around the table. That's not the right direction, I understand. Okay. Can you do this again then?
We use the same strategy to use the commercial libraries that we have got a good collaboration with these commercial vendors that we very quickly can get hundreds of compounds. So we are working on cancer projects together with other researchers at SciLifeLab, where we have developed compounds in the same number with the same speed. And we also got them more aware of the sort of how we can move those to the next step. So we think a lot more about the potential to make these drugs. And we've also received funding to work on other viruses or other enzymes in the virus, which are ongoing right now. Some more challenging, some easier, but certainly the strategy we used here could be used again.
Questions from pandemic preparedness. Oh, Anders Olsson. Raised in hand. Do you want to ask a question, Anders?
Yes, please. Yes. Thanks so much. A great presentation. So, Jens, having the structure information is clearly crucial. If you had only AlphaFold structures for this, would you have succeeded?
That is something we're working on very intensively, and there's been some skepticism on AlphaFold. Recent publications show that they might perform worse in structure-based drug design. We don't agree with this. We think that the AlphaFold models is a remarkable breakthrough and will enable drug discovery for many targets. You can't just download the model on the internet. You have to work with it, which is not a surprise to us because that's what we've been doing all along with different models. So, but it depends a little bit on the target. But for the main protein, I think we believe in learning by doing. So we're carrying out projects now with AlphaFold models, and hopefully early next year we can get back to you on that question.
But it's not time for the crystallographers to retire just yet then.
No, I think the term at the different phases, it's super important to get a crystal structure of your complex, especially in optimization, when really small difference make a big difference in optimization. So for early discovery, I think AlphaFold models could be useful, but in the hit to lead optimization, the crystallographers or cryospectroscopists don't have to worry. We will need them for a long time. And to be honest, which I think sometimes is forgotten in the discussion, is that honestly, even if we get a crystal structure sometimes, we can't design a better compound. So we need both very high resolution structures and better computational methods to predict affinity. So none of it is enough. But AlphaFold width, I think, will be a breakthrough. And if you haven't seen it, there are now posts on the internet where DeepMind shows that they now have extended it to small molecule complexes, which is exciting and scary at the same time. It looks very interesting.
Great, thanks.
And we have a question from anonymous participant. Nice talk. Can you increase potency further by making it covalent? Have you went with compound 19 to clinics with it? 2 questions.
No, so the first question, I think we can probably design a covalent compound based on compound 19. We had some ideas on that during the project, but we have not explored them in detail. We thought that it was actually good for us to work on something different than pharma was doing, so we somehow intentionally have avoided it. And compound 19 is not a suitable compound for the clinic based on poor ADME properties, so it will not go there. But it can be a good tool compound.
Then we have a question from Fong Lam. Do you think that large-scale molecular docking can work for the discovery of inhibitor of protein-protein interaction, as there may be some induced fit conformation changing of protein once binding to their corresponding protein receptor? What is your opinion on that?
So the success of virtual screening depends a lot on the nature of the binding site, and the protein-protein interfaces are often quite flat, and it's difficult to find a good anchor. So I wouldn't expect the same success rates. So here, about maybe 5% of the molecules that we predicted bound. When we work on G-protein coupled receptors, about 30% bind to their receptor. Because those have really nice evolved binding sites for small molecules. So for protein-protein interface, I think it's possible, but one have to also accept a lower hit rate. So you would have to test a larger number of compounds. But we should remember that, if you do high throughput screening, your hit rate is well below 1%. So the computational chemistry certainly helps to get a compound faster. But I believe that for protein-protein interfaces, you might have to have a good budget.
Okay, are there any more questions?
I had one more.
Go ahead.
So the supercomputer, you did this in the initial screen in one day. Will it be better with even larger computers, or is it good enough?
So the big problem for us is the accuracy of the scoring function. So we score a molecule in a fraction of a second, and we know that we make many approximations. So once we get faster, better computer, we will be able to improve the accuracy of each prediction, and hopefully thereby be able to test fewer molecules. And the libraries are growing very quickly. So I said there's about 30 billion molecules available now that we can have on our desk in 4 weeks after we order them. But, and I shouldn't say unfortunately, the truth is that they can make the libraries as big as they want. It could be 1 trillion molecules that we can order. So the libraries are way bigger than we can screen already. So we need to develop new methods. We have one method now that we have developed based on where we use the machine learning AI to try to predict where to go in the big chemical space, not just screen all of them. We will never win. When we have 10 to the power of 60 molecules that we can make, we still won't be able to screen them. So we need clever ways to travel in the chemical universe. And that's what we're working on now.
So will I eventually be able to do this on my iPad?
Yes.
Okay.
I don't know if you or I will be alive at that point, so it might have to be more like on a different in heaven, probably.
Okay.
It's there.
Okay, that's a good final remark then for this project, or I don't know. I think that we are closing now the presentation part of this. event, and we will move down to Mötestorget here in Navet in Uppsala, and there we will hand over the exit documentation from the platform to Jens and Helena and Anja and Lindon, which marks all the work that has been done at the DDD and give you all the data, so to speak. Thank you very much for your talks here. It was very interesting to hear about it, and I wish you good success in the future with your venture with a pharma company.
Thank you very much.
Yeah.